an SME meeting, and then she came here and gave a talk, and that's how we met there first. And we have been fortunate to have collaboration with each other for many years now, published so many papers together. That's good. And today she's going to talk about mutant fixation in the presence of a natural enemy and other stories. Thank you, Natalia.
Thank you so much. So this is my fourth time within 7 days speaking here. Steve has heard all my talks, so I made a new one. So this has never, so the quality may not be optimal, but okay, I try. So today I'll talk about something different. And the motivation for this work is phage therapy and antibiotic resistance. So phage therapy is a form of treatment that uses phages, which are viruses that infect bacteria. And so these therapies are used to combat bacterial infections. And as you know, resistance to antibiotics is a very big problem. So phage therapy is thought to provide an alternative to antibiotic treatment. It was invented a few times. The first time was in 1896. Something in the waters of the Ganges and Yamuna rivers in India was found that provided an antibacterial effect. Then in 1915, it was rediscovered in London, a small agent that infected and killed bacteria. And independently in 1917, a French-Canadian microbiologist discovered an invisible antagonistic microbe of that could kill some bacteria. So although overshadowed by antibiotic treatment, it provides a new hope, so to say, to treat problematic infections that may be resistant to antibiotics. So the way it works is that these phages, these viruses, are highly specific to certain bacteria, making them precise tools for targeting and destroying bacterial pathogens. They attach themselves to bacterial cells. So this photo is actually showing a bacterium which is being attacked by phages. And they use the bacteria's machinery to replicate. And the new phages then burst out of the bacterium, killing it and spreading to other bacteria. So there are some advantages to this treatment, specificity, and perhaps overcoming resistance. But there are also some problems that have been noticed. So in order to model this type of treatment, I'll give you a very simple equation. So if you think about bacteria and viruses, this is a predator-prey system. So here, S is susceptible and I is infected, or this could be the prey and this is a predator, or most generally, this could be a victim and this is an enemy. So there's a predator-prey system, there's a host infection, host pathogen system, or enemy-victim system. All of them have different applications, but mathematically they're similar. And the main term is this one. This is an example of an infection or predation term. It comes out of here and goes here, right? And the predators die, and the victims or the bacteria grow, in this case, according to a logistic growth. Very simple system, but and studied to death, of course. We know that there are oscillations in such system, predator-prey, Lotka-Volterra, everybody knows that. But once we put this stuff in space, right, it becomes very complicated, very complex. So I'll present here an ABM algorithm that deals with such systems. So we have a square grid, and we have bacteria or uninfected cells, and we have infected cells, and cells divide to one of their 8 neighbors, and infected cells can infect one of their neighbors. So it's a usual cellular automaton or ABA algorithm that kind of models such things in space. And one known result when it comes to space is this. If we consider this system of ODEs, there could, for some parameter regimes, there could be wild oscillations. And in the stochastic system, if you go up and down, you go too much down, you're extinct. So such systems are considered unstable for certain parameters, and they lead to an extinction. And once you put it in space, they no longer go extinct because it's spatially distributed. Somehow they survive. So this is a well-known effect of space that stabilizes these systems. Say again? The spatial effect is not in the ODE. So in order to incorporate it into your system, there are several ways. One of them is to write this as a PDE by adding a diffusion term. Or what we do is consider something like this, which incorporates spatial effects. Right? So no, it's not written down here, but this is very well understood. What is not understood is the opposite effect. So sometimes, for some parameter regions, you can have a stable coexistence for a mass action system. So for the system of ODEs. But when you add the nearest neighbors interactions, it drives it extinct. So in this case, space does what it's not supposed to do. It just destabilizes the system. It makes it go extinct. So this is an older project that I wanted to put here just to show you some of the unexpected complexities of predator-prey in space. And then I'll move on to evolutionary problems and phage therapies. Okay, so first let me explain this. This is a numerical result which compares the system in mass action and nearest neighbors. The horizontal, the horizontal axis represents the infection, the reproduction rate of bacteria, and this is the death rate. So this is a two-dimensional parameter space, and each point corresponds to one of the three possible outcomes. So you run the system, and you see, does it go extinct? If it goes extinct, it's red. Blue is coexistence, so it's a stable system where both predator-prey are there. And gray is persistence of host without pathogens. So the virus dies out and uninfected cells are there. So three possible types of outcome. And in mass action, this is the map of what's going on. And in nearest neighbors with the same parameter values, we have the region of coexistence or stability that is smaller than what is observed in mass action. So in this whole area of space, and this is a large area, this is log scale, we have extinction in the spatial system and coexistence in the system of ODEs. So that's a little strange. And so we can, these simulations were performed for a 30 by 30 grid, increase the grid. So again, we have a region which in the spatial system results in the crash and the extinction, while the ODE is happily persisting. So let's take a look of what happens in the spatial system. In this region between these two lines, we have the predicted level of susceptibles or bacteria or prey greater than 1. And this is a spatial picture. So we have kind of more or less uniform distribution of infected and uninfected cells. If we now move here, blue region, but below this white line, we have this very kind of interesting, more interesting landscape where we have islands of cells, and they kind of move around slowly, relatively slowly. So they're large macroscopic structures that are forming in this case. And they kind of chase each other. So the infected cells chase uninfected cells across the space. We don't observe this in a smaller grid because there's not enough space for that. But as soon as we increase the grid, this happens. So it's still coexistence, but kind of like a shaky coexistence, right? And then here, over here, we have extinction. So this here shows you the region where in the spatial system it just goes extinct, right? Spatial interactions destabilize the system. And this shows, again, some simulations where we plot the time to extinction as a function of migration. Very large migration corresponds to non-spatial system. And the small migration coefficient corresponds to the tight spatial interactions. And time to extinction goes to infinity, more or less. Very long time in the absence of space. So in order to model this, we're kind of inspired by this picture where we have macroscopic structures, right? So we can model it as a metapopulation dynamics. So we're going to take these equations and write them as a system of ODEs for different patches. So this is a mass action system where we have one large patch, and here we have a bunch of smaller patches with migration between them, right? So this is a third way actually to incorporate space. So you can do it as a large system of ODEs, each corresponding to a different patch. This is called metapopulation, a DEM model, or a patch model, depending on who you talk with. So this provides often a way to approach complicated problems analytically, which I will show you later. And also another advantage of this approach is that we can kind of generalize it and consider more general types of enemy-victim interactions. Remember, I showed you a simple system here, which has specific forms for the growth term, logistic growth, infection term, just the product. Here, we do it more generally. We say that this is a function f, this is a function g, and this comes back here, right? So this is it's a more general system of enemy-victim interactions, and we have simulations for a couple of these. So this first term, the growth term, can be the standard logistic model, or it can be a different law like this, which corresponds to some situations in ecology. And this term, infection, can be like it was before, just x times y is divided by k, or it can be something with saturation. So let's combine them in all different ways. And you can model that by using metapopulation models. And we still see the same effect. You see, we have to look at this region that is marked by white arrow. This is a huge parameter region where in a spatial system we have extinction, while in the non-spatial system we have coexistence. So it works for many of these systems. We derived some conditions, sufficient conditions that tell you when we can observe that, depending on the form of these functions. This is called the predation function. This is the growth rate. And so the conditions are spelled out here. And there are a couple of examples that have simplified conditions. And they're very simple. So there are some simple conditions, which are just conditions on the partial derivatives of these functions with respect to the variables at the equilibrium that tell you whether or not space is going to destabilize the system.
If you have a system, there are two kinds of interactions. If you are controlling the derivative, this is called competitor cooperative system. There's some similarity with monotone system.
Yeah, that's possible.
And then the completely describe the behavior, one-time behavior.
Yes. So what's it good for? I'll show you some experiments that can show you what it is trying to explain. These are experiments that were conducted for this system of predator and prey. So at the bottom, there are bacteria. They're eaten by ciliates or flagellates, larger organisms. And this is a large ciliate at the top of this. That's one of them. So these things kind of eat smaller things. And in this paper, experiments were conducted in a large reservoir where they all are kind of, this is kind of like your mass action system, or you can look at a fragmented system, which has 4 compartments or 16 compartments. And they can be completely separated, or they were corridors which kind of almost reproduce this mathematical model of meta-populations with ODEs describing each with some migration in between, right? And what we see here is the probability of extinction is the largest for the fragmented or spatially distributed population compared to this mass action system. So this actually happens in nature and was confirmed by some experiments. So this was just to show you that there are some unexpected effects of space. And now I want to add even more complexity and talk about evolutionary dynamics that is observed in these systems. So we are going to add evolution to this and talk about mutant fixation. So remember, that's if you were here for a previous talk, what's fixation? You have, in the simplest Moran process, two types, and one is killed and the other one reproduces, and you wait until the mutant, which has different properties, takes over or goes extinct. So depending on the fitness of the mutant, the fitness is 1 plus S, and we call S the selection coefficient here. Depending on that, the probability of fixation can be large or small. When S is equal to 0, it's a neutral mutant, and the probability of fixation is known. And S positive means advantageous, S negative means disadvantageous. Everything is known about this Markov chain, and you can calculate this probability. Now, we are going to do the same in space. Probabilities are similar. And even in a contact process where you have some demographic fluctuations, probability is still similar. But next, we are going to introduce infection. So all these problems were solved for systems that consist of two types, wild type and mutant. What's the probability of fixation of a mutant? Okay, this is all very well understood. But in nature, nothing exists in isolation. There are always predators, there are pathogens, there's infection. So all these systems are subject to natural enemies. So how do natural enemies influence the course of evolution? So that's what we're going to check here. We're going to put infection in the system and calculate the probability of fixation of a mutant. So here, these 2 points, this point, for example, corresponds to the absence of infection. And then we look at systems with infection and increase kind of the intensity of infection or the intensity of consumption of these species. And you can see that for an advantageous mutant, the probability of fixation decreases. So the more infection or the more predation you have in the system, the less advantageous is that mutant, although that mutant has exactly the same properties. So it's the same system, it's just being eaten by somebody. So somehow the probability of fixation decreases. Now we take a disadvantageous mutant. In the absence of predation, it has this probability of fixation. Now let's put a predator on it, and it increases. So for a disadvantageous mutant, it becomes less disadvantageous. The probability of fixation increases. So what we see is that advantageous mutants are less likely to invade, disadvantageous mutants are more likely to invade. So selection is weakened by infection. So that's an interesting effect, and we try to understand it mathematically. So let's look at the dynamics of uninfected cells as a function of time. They oscillate, okay? But if we look closely at the spatial distribution of infected, uninfected, and empty patches, we see these macroscopic structures again. And this is what, again, inspired the mathematical model. So we're going to look at the metapopulation again and write down ODEs for each of these metapopulations with migration in between. Right? Computer simulations show that in this case, we have exactly the same effect. The advantageous mutants in the presence of infection are less likely to invade. Disadvantageous mutants are more likely to invade. So in the system of metapopulations, we have the same effect. So it's a good model to look at the space. When we look at the overall dynamics, it looks like oscillations. Look closely at one patch. There are extinctions of the population that happen periodically. So the overall number of species never goes to 0, but inside each patch, they crash all the time. So this led us to considering a dynamics in patches, which we call a coarse-grained dynamics. We look at patches that are completely, they completely consist of normal cells, completely consist of mutant cells, Y, and these are infected patches in yellow. So I think I'm running out of time. We are going to consider, instead of looking at specific individual cells and their enemies, we're going to have a different spatial scale where our unit is a patch, a fully wild type patch, a fully mutant patch, or an infected patch. First, we did it without mutants and derived a coarse-grained equation like this. And this led to results. So I have to skip all of this. That the results coincided very well with simulations. So the coarse-grained patch model kind of reproduced the overall properties of the detailed microscopic model. And then we added mutants. So now we have the following processes. We have patches that are completely wild type and uninfected, patches that are completely mutant and uninfected, and we have patches that contained some infection. So these are going to die out. The infection is going to crush them. And then they can get recolonized. An empty patch can get recolonized by either wild types or mutants. And there's also a conversion between these. If you put a mutant in here by migration, it can kind of take over, or the other way around. So there are several, so there are all these processes that take place on this coarse-grained system. And the good thing is that we can calculate the rate of all of these analytically. So this shows you that this is a good approach, because in the stochastic simulations, we can see that most of the time, the patches are completely mutant or completely wild type. There are almost never patches that contain both types of the population. So it's a good approach to kind of just consider homogeneous patches. We can calculate the rates of colonization, conversion, infection, and conversion back. This gives us an analytical expression for the selection coefficient of a patch. So it's easy to calculate a selection coefficient for an organism, but this is a selection coefficient in the population of patches. And we have two regimes. So first of all, the predictions of this approximation very well coincide with the simulations, but here's an explanation that it provides. So we have two processes, extinction and recolonization of patches and conversion of the patches. If this conversion is the main process, then the selection coefficient of the patch is the same as the system in mass action, so there is no change. But if it's the other way around, if there are macroscopic structures, a lot of empty space, and the main process is extinction recolonization, then selection is very much weakened, because the selection coefficient of these patches is a lot smaller. The absolute value of that is a lot smaller compared to the original system. So why is this? What does it have to do with phages and treatment? So this is a paper that considered bacteriophage interactions and the selection dynamics with and without phages. So without phages, bacteria are competing with each other, and the fittest species will win and just kind of stick around there. In the presence of phages, selection is weakened, which means that some of the weaker subtypes of bacteria can stick around for longer. There are lots of them in this fitness landscape. And these are perhaps the ones that will later exhibit resistance to antibiotics, right? So here we have only one type, the wild type of bacteria, and you can treat it with antibiotics. If there is this variety of different types that are hanging out because selection is weakened, now you start treating with antibiotics and some of them will be resistant. So this is kind of a negative view of the phage therapy, because although it can treat the disease, it may have an unwanted side effect where it actually creates conditions for antibiotic resistance to come up. So this is an unexpected consequence of this type of treatment, and it's now being noticed in the literature that this indeed may be the case. So I wanted to thank my group and Dominic, who collaborated with me on this. And I came now to the most important slide of my talk, which has to do with the reason why we're here. I want to read the following poem. In Waterloo, where minds are bright, he crafts equations with insight. Through math and life, he is there to give a guide, a friend, they call him Sieve. From biofilms to cancer spread, he solves the puzzles in his head. In hearts and minds, his work will live, a lasting legacy from Sieve. We're here today to celebrate a man who is kind and true and great. There is simply no one better than our dearest Dr. Sivalloganathan. And I want to present Siv with a little, this is basically this poem that you can put on your desk.
If you want to pick it now, or should I give it to later? You want to pick it now or later? Yeah, maybe.
Yeah.
Yeah, maybe. Yeah, come.
I think we're going to make you poet laureate. Anyways. Thank you.
Okay, wonderful. Now time for questions.
Maybe I can ask a question while I'm-
Sure, yes.
On the contents of the poem? Is
No, no.
something wrong?
No. It's sort of interesting, and a bit counterintuitive. What you said about the fixation probability and the advantageous mutants. So I was just trying to think, what do you think the explanation is there? That the infection targets more the advantageous mutants because they're proliferating faster.
So it goes both ways. It makes this advantageous mutants less advantageous, and it makes disadvantageous mutants less disadvantageous.
Yeah, because what attacks in that case the wild type because they're proliferating.
So it has to do with the fact that in this regime, a lot of patches, a lot of space is empty, and it gets recolonized by cells from other patches. And while they are recolonized, they are not competing with each other. So that's the intuitive explanation. If they were always all competing in each patch, then of course, if I'm a disadvantageous mutant, I feel, But if there is an empty patch here and I'm a disadvantageous mutant, I jump here and there is nobody around me. And I grow without opposition. So that's an intuitive explanation. And it only works in the regime where there is a lot of infection going on. Infection creates empty patches. It creates empty space. And that's where they don't compete with each other. So.
Okay, thank you. Any other question? Okay, maybe one more.
So this conclusion you put at the end regarding with the presence of phages, then it may, under some conditions, make it worse. Did you look at just one type of phage? Because as you know, some phage therapies does involve the mix of several phages together, not just one type of phage. So what about the fitness of the phage itself?
So you're raising an important question. So this model only takes into account one phage, and we assume that the behavior of an infected cell is the same. So everybody is equal in the face of an enemy. So that situation gives rise to the effects that I described, and it basically increases the evolutionary potential of the bacterium. They are more likely to hang around and create a larger variety of mutants, a larger degree of diversity. Now, if we add more phages or include the co-evolution of phages and bacteria, that may bring other effects, and that would be the next step.
Okay. One more